Earlier in this chapter, I mentioned that bacteria can undergo horizontal gene transfer. So two bacteria in the same culture can actually exchange genetic material. This requires that the two cells are in close contact. This is not going to happen with one cell and then with another cell that's on the other side of the plate or the culture. They would have to be in direct contact. This is what we call conjugation. So they would actually mate where the bridge would form and genetic material would flow from one bacteria to the other. I often use the analogy of conjugal visits in prison, so you know there has to be direct contact. So that's why I often use as an analogy. So in order for bacteria to transfer genetic material, there has to be a passageway. And this passageway is a conjugation pilus or conjugation bridge. There's a couple of different names for this. This is an F pilus, sex pilus, or pilus, however it is that you'd like to say it. And the directions to making this conjugation pilus reside in a fertility plasmid or an F plasmid. So if you remember from the beginning of this chapter, I mentioned that plasmids have nice to have genes. These are not necessary genes for the survival. This is like extra DNA. Not all bacteria have plasmids. If a bacteria has an F plasmid, a fertility plasmid, it has the genes to direct the synthesis of this conjugation bridge. And that's how genetic information can flow from one bacteria to another. So if we have a bacteria and it has an F plasmid, we would call that the F plus cell. That's the donor cell, which will donate DNA. The recipient is going to be the F minus cell. Now, another term they'll use is male and female. I mostly use F plus in donor and F minus and recipient. So in a culture, if we have a bacteria that has an F plasmid and it's an F plus, it's a donor cell, eventually, all of those cells are going to become F plasmid cells because one bacteria is going to send genetic information to the next. Then that cell will become an F plus cell, and then that cell will donate genetic material to the F minus cell, and that cell will become an F plus cell, and so forth. So if we wait long enough, all of those bacteria in culture will actually become those F plus cells. So I mentioned that the genes to construct this conjugation bridge are going to be found on plasmids. So remember, not all bacteria will have plasmids, and these fertility plasmids have the genes, the instructions to make this conjugation pili. Without that, genetic information is not going to be able to pass from one bacteria to the next. I also mentioned that plasmids can contain nice to have information. Some of this information could actually be resistant genes. And so we're going to spend a little bit of time talking about resistance. Resistance means that an antibiotic that once killed a bacteria now can no longer kill that bacteria. That bacteria is now resistant to that antibiotic. So if you take a look at this picture, this is a picture of bacterial conjugation. So on the left-hand side, we've got our donor cell, our F plus cell. It has a chromosome, and you notice that it has an F plasmid. That F plasmid has the instructions to make that conjugation pilus. So now that conjugation pilus forms, remember that's something unique to bacteria, and it can actually exchange genetic information with that F minus cell, that recipient cell. So once that conjugation bridge forms, now some DNA can actually transfer. In the case of this bacteria, the plasmid, a copy of the plasmid, is actually being transmitted from that donor cell to the recipient cell. And now at the end of conjugation, when you look at that bottom picture, those two cells are identical. So now that recipient cell has now become an F plus or a donor cell. So I keep mentioning this resistance. So I want to talk a little bit about resistance and what this means. We can actually test for antibiotic resistance in the lab. In fact, we're going to be doing that coming up in an upcoming lab. If a bacteria is resistant to an antibiotic, that means that it has these mechanisms that work around that antibiotic killing it. We're going to get into the specifics of those mechanisms coming up. But if we had a bacteria and it had a resistant gene and we grew it in the presence of that antibiotic that it had a resistant gene for, it would actually live. There's nothing that would kill it. That antibiotic is not going to work on it. So it would live, it would grow, and we would probably assign a little plus to say, yes, there's growth. If that bacteria is susceptible, now it doesn't have a workaround from that antibiotic killing it. So it's susceptible. It would actually die. We would not see growth on that plate if we plated this organism in the presence of that antibiotic, and it would die. We would put a minus sign. So to find out if a bacteria has a resistant gene, we could actually plate it in the presence of that antibiotic. If it has that resistance gene, it will live. If it does not have that antibiotic resistant gene, then it will die. Just a little note that I want to make sure the vocabulary is correct. It's not that the antibiotic is resistant, it's that the bacteria is resistant to the antibiotic. Sometimes we end up confusing those, but it really is the bacteria that has a workaround that antibiotic killing it. Again, later on, we're going to get into those specific mechanisms. So a little example here. So I have 2 strains of E. coli. So if we were to look at their biochemistry, they would type out as E. coli, but you notice that they differ a little bit genetically. This is why they're strains. So strain 1 has a chromosome, obviously. It has to have that to survive. And it has a streptomycin resistant gene on that chromosome. If you take a look at strain 2, it has a chromosomal gene, of course, and it also has a plasmid. You notice that strain 1 does not have a plasmid. So on the chromosomal genes of strain 2, it has a now resistant gene, meaning that it would be resistant to that particular chemical. On its plasmid, it has an ampicillin resistant gene. So there's a media that if we're testing for resistant genes and conjugation, we actually use an LB agar. And this is a special agar that allows the bacteria to kind of open up and remain healthy. So that's a type of media that we would use. So if we were to actually plate these 2 bacteria in a variety of situations, this is actually what we would see. So if we were to plate our strain 1 in plain agar, it would actually grow. There's nothing in that media to kill it. So I'm going to put a little plus sign there. If we were to plate strain 2 in that plain LB media, there's nothing in it to kill it, so it would grow. So we would see growth. We would note that with a positive. So this is just to show we want to do a control, just to show that this bacteria is growing. It's able to use the nutrients in that media. Now let's take a look at a situation, that second row, where we have the media, but we've added ampicillin to the media. So strain number 1, would strain number 1 grow? If you notice, it has a streptomycin resistant gene. It does not have an ampicillin resistant gene. No ampicillin resistant gene, so that ampicillin in the media would actually kill it. So we would not see growth. I'm going to put a little minus sign there. For strain number 2, would that grow? If you notice that strain number 2 has an ampicillin-resistant gene on the plasmid, that ampicillin-resistant gene is going to give it a mechanism around that ampicillin killing it, so we would expect growth. So I'm going to put a little plus in that column. If we look at the third situation, we've got the media with a streptomycin added to the media. If you look at strain number one has a streptomycin resistant gene, so it's got to work around from that antibiotic killing it, so we would actually see growth. If we look at strain number two does not have a streptomycin resistant gene, so it would die. We would not see growth, so I'm going to put a minus there. Now, let's kind of combine antibiotics and see what happens. So if we plated strain 1 in the media with both streptomycin and ampicillin in the media, it would have to have both of those genes to survive. So strain number 1 has a streptomycin resistant gene. It does not have an ampicillin resistant gene. The streptomycin in the media is not going to kill it, but the ampicillin will. So we would not see growth because the ampicillin would kill it. If we look at strain number 2, there's an ampicillin resistant gene, but no streptomycin resistant gene. Ampicillin in the media is not going to kill it. The streptomycin will. So again, we're not going to see growth. If we look at the case of NAL, strain number 1 does not have an NAL resistant gene, so we would not see growth. If we look at strain number 2, it does have an NAL resistant gene on its chromosome, so it would have that workaround, so we would see growth. So we would put a positive. So this is what we would call a confirmation, just to find out what genes we suspected our strains of E. coli had. So if we take a look at a picture and how those genes, those resistant genes, can be transferred, if we have our donor cell, our F plus cell, it has that fertility plasmid. It's next to that recipient cell. It has that F plasmid, so it can have the directions to make that conjugation bridge. Conjugation bridge forms. And now, if you notice, that F plus cell is actually transmitting a copy of its plasmid. So now at the end of this conjugation and procedure, that F minus cell looks exactly like that F plus cell. Now it's become the donor cell. Now, if let's say that F plus cell has an ampicillin resistant gene on that plasmid, now that cell next to it that was a recipient now has that ampicillin resistant gene. So when we talk about resistance and what an issue resistance is and how resistance can spread among a population, this is exactly how it can occur and why it can occur so quickly, because conjugation can happen. These donor cells can then transmit a copy of their antibiotic resistant gene. So this is a big issue. So here is an experiment that we actually did in lab. And on the right-hand side, I actually have like what our strain would look like. And so if we grew a strain in lab and we wanted to find out if it was resistant to certain antibiotics, certain chemicals, we would grow it in the presence. If we see growth, we know it has that resistant gene. If we don't see growth, then we know it does not have that resistant gene. So this is actually a case where mating happened, conjugation happened. So I gave you that example of strain 1 and strain 2. So strain 1 and strain 2 mated. And so what happened is that a brand new organism was made. It has a different genetic map, so it's a different strain. So if we take a look at what strain 3 will actually grow on, this can give us an idea of what its chromosomal map looks like. So if you take a look, strain number 3 grew in the presence of ampicillin. So does that mean that it has that ampicillin resistant gene? Yes, it does. It has that ampicillin resistant gene. So I'm just going to put a little A right there for our ampicillin resistant gene. Now we grew it in the presence of streptomycin. Does strain 3 have a resistant gene for streptomycin? Yes, it does, because we see growth. So we know that it has a streptomycin-resistant gene and an ampicillin-resistant gene. Now, does strain number 3 have a NAL-resistant gene? If you take a look at that plate where there's no growth, it was actually growing in the presence of NAL. There's no growth. It does not have that NAL-resistant gene. Our last scenario, we plated our conjugate, our strain 3, in streptomycin and ampicillin. Remember, in order for it to grow in the presence of both of those antibiotics, it has to have both of those resistant genes. And it does. If you look at our chromosomal map, it has the ampicillin-resistant gene and the strep-resistant gene. We found that out by plating them in those separate situations. But when we combine them, you can see that it's got to work around for both of those antibiotics. So our strain 3 has an ampicillin-resistant gene and a streptomycin-resistant gene. And this is how we can tell if a bacteria has a antibiotic resistant gene. So another thing that can happen is that the plasmid can actually incorporate itself into the chromosomal DNA. This is what we call a high frequency recombination, or HFR. Recombination means that we're combining DNA. So that plasmid is going to meld into the chromosomal DNA. So if you take a look at this picture, we have our F plus cell at the top with an F plasmid. And in that next picture, that F plasmid becomes incorporated into the chromosome. So now it becomes an HFR cell. This HFR cell is still the donor cell. So now it can mate with a recipient cell, and some of those chromosomal and plasmid genes can actually move. So I know we've been talking about plasmids. So sometimes we get that idea in our head that, oh, only plasmid genes can move. No, chromosomal genes can move. Plasmid genes can move as well. If we see a case of an HFR, you can see where the combination of those plasmids and chromosomal genes can actually be transmitted to that recipient cell. This is often what we call jumping genes. Now, sometimes that plasmid actually comes back out of the DNA, and it might actually take some of that chromosomal DNA with it because it looks a little different. We go from calling it an F plasmid to becoming an F prime plasmid just because it looks a little different. So we have to give it a little different designation. So here's my drawing of our HFR cell. So it's got that circular chromosome. It has that red plasmid, and then that plasmid becomes incorporated into the DNA. And now we have an HFR cell. If that plasmid comes back out, it's not going to be a perfect excision. It's going to end up taking some of the chromosomal genes with it. And now it becomes an F prime plasmid. If you look at that, it's got the red F plasmid genes, but it also has a little bit of the blue, the chromosomal genes. So because it looks a little different, we have to give it a little different designation, which is why we call it an F prime. So again, this is just part of that scrambling and how genes can kind of be mixed up. So here is our HFR cell, and then you can see where that plasmid comes back out. And now it's an F prime plasmid. So now we've got our F prime donor cell, and it donates a copy of its plasmid to a recipient cell. Now that recipient cell looks the same as that donor cell, but now it has the benefit of whatever chromosomal genes were there. So if, let's say, there was an ampicillin resistant gene on the plasmid, and by that high frequency, it actually pulled out a streptomycin resistant gene. Now we're actually donating 2 resistant genes instead of 1. So this is what we often call jumping genes. And jumping genes is the ability of these genes to move. These genes can move from plasmid to plasmid. They can also move from plasmid to the bacterial chromosome. The ability to move from one location to another is what we call transposition. And this is made possible by these transposable elements. This is often what we call jumping genes. It can jump from one thing to the other. These transposable elements actually have a genetic sequence on the DNA, but then there's also the codes for an enzyme, transposase, on the ends, kind of like bookends of these transposable elements that allow it to actually be excised out so that it can actually move from one place to the other. So if you take a look at this plasmid, it has this transposable element. So on the middle, we've got our transposon genes, and then we've got those inverted repeats that are kind of like bookends of these transposable elements. There could be an antibiotic resistant gene on this transposable element. If it moves from plasmid to plasmid or plasmid to chromosome, then it's going to take those antibiotic resistant genes with it. So this is a good way to see how resistance can arise so quickly with bacteria. We're going to be talking a lot more about resistance coming up in the upcoming chapters. And it also kind of gives you an idea of how these bacteria can increase their genetic diversity and kind of do that scrambling of genes.